So it's a pleasure to have with us an invited Josep Carreras Institute lecturer, Dr. Pancho Barriga, Francisco Barriga. He was, I don't know, born by accident or not in Washington, in the US, but at 5 years old he moved back to Chile, where he got his biochemistry degree at the Pontificia University Católica de Chile. And from there, he moved to do his his PhD at the lab of Eduard Valle at the IRB here in Barcelona. And there he works on stem cell heterogeneity in intestinal stem cells. From there, he moved to do his postdoc at the lab of Dr. Scott Lowe at Memorial Sloan Kettering Cancer Center in New York, where he developed new approaches to study copy number alterations in cancer. And he identified a particular deletion that was associated with intrinsic and extrinsic tumor suppression. He's currently, very recently, the Group PI and leader of the Cancer Genome Engineering Lab at the Vall d'Hebron Institute of Oncology VHIO in Barcelona, where he continues his studies in immune evasion, tumor heterogeneity, and cancer genome evolution. So it's a great pleasure to have with us Pancho. The floor is yours.
Oh, a rare pre-lecture clap, thank you. So first of all, thanks so much, Manel, for the invitation. This is literally my first talk as a group leader. I was a postdoc until, I don't know, 2 months ago, so I don't know if it'll change. And I am, like, half of the lab is here, so my lab manager is back in the lab fighting with customs to get our cells from New York into Spain. So today what I'll try to do is basically give you an overview of the work I was doing in Scott Lowe's lab during my postdoc. And as Manel mentioned, it's centered on this, trying to understand what copy number alterations do in cancer. Now, this is a very big question. I don't think we answer this, but I think we're starting to build a framework where we can systematically try to understand what these large-scale genetic events do. So just to introduce this very briefly, and this is the most simplified cartoon I have that I always use, the transformation of a normal epithelial cell into cancer is through the genetic activation of oncogenes and the genetic inactivation of tumor suppressor genes. Now, collectively, the cancer genetics field has been extremely successful at the identification of genes that are altered by point mutations. So specific genes that have single nucleotide variants that are either activating or inactivating their function. Now, we also know that cancer genomes, although they are very much filled with single nucleotide variants, are not just that. If you look at a cancer genome, there's actually a lot more going at a structural level. And I'll just exemplify this as follows. So here on the left, you can see a normal epithelial cell, and this is a sparse whole genome sequencing. So here on the x-axis, you have the genome lined up by chromosomes. In the y-axis, you have the dosage. So as one would expect, a normal cell has 2 copies of each chromosome. Now, when we look at our cancer counterpart, and this is actually a myeloid leukemia, complex karyotype, we see that this pattern is deviating very much. Now, here in blue are regions that are lost or deleted, and in red are regions that are gained or amplified, and we collectively call these changes copy number alterations. Now, again, this is not new. We have known this for a very long time, but I do think that the field has been somewhat simplistic in its vision of how these actually work or how do they impinge on cancer function. So arguably, this gain of chromosome 8q, if you go to most papers, it would say, well, MYC resides here. So the entire effect of gaining 8q, well, that's probably just MYC. Although there are around 600 genes that are also co-amplified, it's just MYC. So I think that although MYC is absolutely relevant, I think there must be much more to the biology. One of the things that I find very interesting of copy number alterations is that they have recurrent cancer-specific patterns. Because there's a valid assumption that through chromosomal instability, this would be just stochastic events. But the more and more patients we sequence, we see that there are recurring patterns that show up in cancer-specific manners, and that also have clinical correlates. Now, why am I, as a more fundamental biologist, interested in copy number alterations? Is because their operational logic is different than point mutations. So if we take, let's say, a made-up locus, we'll call this wild type, it has genes A to J. If you have a point mutation in gene A, this will alter the function of gene A, which makes sense. Now, in the case of a copy number alteration, you're not only affecting gene A, you're affecting the whole region. So in the case of a gain, the effect that this gain may have might be through a combination of multiple of these genes. And the same for deletion. The deletion may have an effect on a cancer cell, not by just losing gene C, but a combination of all of these genes. Now, there's a second part that I will not be talking about, but I am extremely interested in pursuing in my own lab, is that copy number alterations do a second thing, which is change what I call DNA topology. That's like a loaded word, but let's just leave it at that, where you actually create new contacts of DNA when you lose a region, or in the most extreme case, you can have oncogene amplification in extrachromosomal DNA. And we have some interest in the lab in understanding some of these structures. Now, I was working with Scott Lowe. He's very famous on tumor suppressors, and tumor suppressors are lost by deletions. So the focus of today's talk will be on deletions. I do have active interest, of course, in amplifications, gains, and so on and so forth. So again, just to reiterate, I'm not saying you anything we didn't know. We have been extremely good in this large repositories of sequencing patients, and we have created now this catalog of copy number alterations across cancer. This has been done by the TCGA, the PCOG, and so on and so forth. And we even have clinical correlates with these alterations. Now, when someone says, let's say, the deletion of a particular chromosome correlates with a particular clinical outcome, this is where we start having issues because we haven't been as good as functionalizing these observations mechanistically. Now, the advent of site-specific or sequence-specific nucleases, particularly zinc fingers and CRISPR, has allowed for a few studies to start doing this. Now, unfortunately, when I started my postdoc, I realized that methods remain limited, and only a handful of copy number alterations have been analyzed. So, how can we actually study the effect, and then here instead of CNAs, I'm going to say deletions, how the phenotypic traits that deletions give to cancer cells? And the answer is not, again, a surprise. Well, we'll delete them, but how do you do that? With CRISPR. So if everyone is familiar with CRISPR, of course, I'm assuming that you understand that when we introduce a guide RNA and a Cas9, you will create a double strand break. And if you introduce two, you will create a second double strand break. In the scenario where these two double strand breaks occur in the same molecule of DNA, there's a chance that the intervening region will be lost. And actually, this works remarkably well. Anyone that's done CRISPR in the lab knows that CRISPR works really well. But when you talk about making deletions, when you start moving from, okay, 100 base pairs, no problem, 1,000 base pairs, easy. 10,000 base pairs, you start getting into issues. 1,000,000 base pairs at 1 megabase, it gets much more complicated. And at the end of the day, what we do is just do single cell cloning. And I did think, like, as much as I like New York, I didn't want to spend my entire time in New York picking clones. So I thought, well, there must be a better way to play this game. And I came up with this approach that I called molecular alteration of chromosomes with engineered tandem elements, or machete. This is reverse engineered. It just didn't pop into my head. And machete is actually it's a very simple concept. Is how do I make a cell with a deletion have a selective advantage in culture? This we do all of this in cell lines. If in the case you had a deletion that you remove a gene that the cells like de facto and they proliferate faster, that would be great. But many times we want to work with regions that have no phenotypic trait in the culture. So what we did is, or what I came up with is, well, how about we just forget about what the sequence is and we just synthetically make these cells sensitive or resistant? So for that, what we do is CRISPR-mediated knock-in of an inducible suicide cassette, in this particular case, diphtheria toxin receptor, and puromycin, which will enable the efficient integration. For everyone that's done CRISPR knock-ins, inserting something into the genome, although it works, is extremely low efficiency. So with puromycin, we introduce this into our cells. We select very strongly with puromycin, and we know that every single cell in our culture has at least incorporated this cassette. And now what we do is we just plug in the standard CRISPR pipeline. We bring 2 guide RNAs to make our deletion, and a fraction of cells, hopefully most of them, will have this inducible suicide cassette in the middle of the region. So what do we do now? There are many possible outcomes. Well, I didn't pick single cell clones at the beginning, so it might be a fraction of cells that this knock-in step went elsewhere, so you'll have an off-target integration. The most frequent is that the guides will create indels, but the region won't be deleted. A rare case would be that you flip this around, and in the case of a deletion, you effectively get rid of this region. Now, the trick here, and I'm sure you see it by now, is that if now I actually give the toxin the only scenario where the cells have an escape route, is in the case where they deleted something. So, and this actually works remarkably well. It's very simple. I mean, the design is somewhat complex, but the idea is just how do I basically kill everything that doesn't do what I want? So I guess the analogy is it's a needle in a haystack. So you could go and look for the needle, or you just burn the hay, and then whatever's left is your needle. So there's a few features that I design on purpose for this. It's rapid and flexible. So if you see, this is 40 base pair homology arms. So what I've done now is I have a collection of these plasmids on add gene. The only thing you need to do is PCR amplify this and just add in your primers, 40 base pairs, and this works. It's easy to adopt because, again, there's no cloning required. You get the plasmids, you can buy the guides or make them, and it's iterative, and I find this particularly interesting. These cells, at the end of the procedure, although they have a deletion, they don't have any of the exogenous elements. And I've shown that we have in supplement something, that we can do this over and over again. So we can start now making increasingly complex genomes because at least we can start making or compounding deletions. So, okay, cool. I spent like a year of my life in a fancy way to do CRISPR. So where do we start? And for this, it was a really good collaboration with Scott because we both share very similar interest in the copy number space. And we were also very interested in this particular locus. And this is chromosome 9p21.3. Now, this is the most recurrent homozygous deletion. Most of the ones I've mentioned, most of copy number alterations that affect large regions are heterozygous. This is, let's say, the exception to the rule. And with clinical prognosis, basically any tumor that has this deletion the patients fare very poorly. So having the 9p deletion is one of the worst cytogenetic events you could have. And we thought we really understood this. This is one of the most studied regions in the human and mouse genome. And it's because there are three major tumor suppressive activities encoded here: ARF, CDKN2A, and CDKN2B, which collectively activate RB and P53. So it makes a lot of sense. You lose this region, you lose these three genes, and you basically inactivate RB and P53, and these cells now have a very strong proliferative advantage. And that is absolutely true. Now, the thing is that all these studies, which are fantastic genetics, were done in the late 90s, before we even knew there was anything else, before we even had the Cancer Genome Atlas. So we were intrigued because there were some observations coming in the literature that didn't fully reconciliate with the functions that we knew about these genes. So we just, okay, let's take a step back and let's look at the patient data. What are the patient deletions looking like? So now I'll show you pancreas cancer patients. This is from TCGA. This is like, I don't know if you're doing oncoprints, it's an oncoprint on its head. So each column is a gene, each row is a patient. The deep blue color means a deletion. And in pancreas cancer, we know the loss of CDKN2A and B is a very frequent event. Now, what happens next to this? So one thing we realize is that there's an adjacent cluster of type 1 interferons that are physically linked. All of this is like 1.5 megabases, 1.4. That also gets co-deleted in a fraction of patients. Now, interferons are very interesting molecules. They're bona fide antiviral molecules. And we were just wondering, well, would there be a phenotypic difference between these two genotypes? Because arguably, the driver event is the loss of the tumor suppressor. This could all just be passenger events. This is stochastic. The deletion will be larger in some patients, more focal in others. Just to remind you a bit about interferons and why this was, let's say, somewhat unlikely. A cell that's infected with a virus will produce interferons and will signal to neighboring cells, either to put up barriers in the form of interferon-stimulated genes or ISGs. In a case of infected cells, they will die, and most importantly, they will recruit immune cells. Now, cell by cell, gene by gene, the cells that produce the most and respond the best to type 1 interferons are immune cells. So removing type 1 interferons from tumor cells doesn't really mean much because the interferons in the immune cells are intact. So arguably, why would this have any phenotype? Again, the data in the literature was somewhat in the air on this, so we just decided to go ahead with it. So how can we test the role of type 1 interferons? And here is where my mouse modeling person comes out and is like, if you want to study anything that has immune relevance, it has to be in mice. Everything else, it's fine, but you don't get a full actual immune response. So the first thing we did is, can we even do this in mice? And the answer was yes. we're fortunate enough that 9p21.3 is fully synthetic to chromosome 4c4, all the same genes, almost in the same orientation. Everything is very much conserved. So the second thing we had at hand is, well, we had machete. So we applied machete in mouse cells. And since our lab has a very strong interest in pancreatic cancer, we decided to take this initially, or most of what I'll be showing today is pancreatic cancer with a few exceptions. And for this, what we do is we take premalignant pancreatic ductal epithelial cells. These are Carreras mutant, and we inactivate a p53. Just for experimental ease, and this came very useful later on, and the next slide I'll show you why, we also introduced the luciferase GFP, just for us to be able to track the cells we engineer. So then we just do and created two alleles, the small deletion and the large deletion. I'm not gonna complicate myself with names. So 0.4 megabases and 1.4 megabases. And this is just a sparse whole genome sequencing showing that we can effectively engineer these deletions very efficiently. And the question is, okay, now we actually have our experimental setup. We have our cells that have two deletions that mimic two different genotypes that we observe in patients. And of course, the question is, do these have any phenotype? So the experiments we do are fairly straightforward. The trickier bit is getting the cells, but now what we do is we transplant them into the pancreas of mice. And the beauty of this is we can transplant it in the pancreas of mice that can be immune competent, immune deficient, and I'll show you why that's important. I'm gonna summarize like 7 figures in just this little plot here. We do have a lot of phenotypes, and I'll just walk you through 2. So the small deletion is more immunoedited than the large deletion, and there's more metastasis in the large deletion. So what do I mean by immunoediting? Is that, as I mentioned, we had actually introduced the luciferase GFP cassette into these cells just for experimental ease. Not surprisingly, most of these mice got tumors, which makes sense. These are pancreas cancer cells that are Carreras mutant and p53, which is usually what a pancreas cancer cell needs to make a tumor. What we did realize is that our small deletion tumors were coming out without GFP. And when we repeated this experiment in immune deficient mice, we could rescue this. So there's something about adaptive immunity. So remember, nude mice don't have T cells or B cells that are functional. There's something about adaptive immunity that is allowing GFP cell, or it's killing GFP positive cells in this small deletion. And we call it immunoediting because we could actually see that those cells with more copies of GFP were being culled by the immune system in our interferon proficient cells. So there's not a lot of functional insight, except there is something related to immunity in our genotypes. So the more interesting phenotype came about with metastasis. So once we transplanted these cells, we realized that the interferon deficient large deletions were extremely metastatic. And in this model that looked in intraintestinal, mesenteric, lung, and liver. And again, we could fully rescue these differences by just changing the host. So far what we've seen is that the loss of tumor intrinsic type 1 interferons with these deletions is doing something to adaptive immunity that is driving tumor progression. Or in the case of the interferon proficient lines, it's engaging anti-tumor immunity. So of course the question is, if the host is the determinant of the phenotype, what is the response in the host? What are the immune cells? How are the immune cells reacting to these tumors? So again, I'm gonna summarize a lot of flow cytometry and single cell RNA-seq. And on average, the interferon proficient cells are much more infiltrated. And we had a very paradoxical increase in CD8 T cells, which are cytotoxic and anti-tumor. Now, of course, that a CD8 T cell is in a tumor doesn't mean it's doing anything. So, of course, what we did is we went and looked deeper. What are the phenotypes of these cells? And what we observed, and this is just a UMAP of CD8 T cells from our different mice, is that although there are very few CD8 T cells, these ones are the interferon proficient, or the CD8 T cells that are in an interferon proficient microenvironment. And this is actually a progenitor-like that is high for effector molecules, and that also has been associated with chronic viral infections. On the other hand, most of the cells that we see that are specific for our large deletion are terminally differentiated LAG3 and TIM3 high cells. Now, of course, this doesn't imply function, and I'm a big fan of that. So what we did is, well, let's just test, are CD8 T cells relevant? Because nude mice have a lot of other issues. So what we did is just took an antibody depletion. So what we do is we remove only CD8 positive cells of our mice, and then we repeat the experiment. And now what we observe is that now our interferon proficient lines are just as metastatic as our interferon deficient ones. And this actually doesn't go higher in the large deletion. So although these tumors are much more infiltrated with CD8 T cells, these T cells are very much irrelevant. It doesn't matter if they're there or not, the tumor just does what it does. Now, importantly, and this is the one thing I do say to my fellow mouse modelers, is this is a mouse, so what's happening in patients? Unfortunately, in a collaboration with Fayaz Nod and Amy Sang at the Ontario Institute of Cancer Research in Toronto, we could actually see that in their PDAC data set, if you segregate patients by large and small deletion, most of what we found in our very much artificial mouse models is conserved in humans. And now, I mean, I'd be remiss not to show at least one slide on therapy, because I do come from Memorial Sloan Kettering. So what we did, and these are proof of concept only, is we used a very potent combination that in the lab we have observed gives rise to response to immune checkpoint blockade in pancreatic cancer. Bear in mind, this has no clinical value. This combination has been stopped because it's too toxic, but our mice are much more able to take it. And this is just a combination of MEK inhibition with CDK4, so trametinib and palbociclib, that if combined in PD-1, we had previously published that elicits a CD8-dependent immune infiltration, immune response. And in our tumors, what we observed, and this is ultrasounds of our interferon-proficient tumors, is that in the presence of the combination plus anti-PD-1, we start getting necrosis. And this correlated very well with the a higher infiltration of T cells and also a more antigen-presenting phenotype of tumor-associated macrophages. Now, these tumors are extremely rough, so although you get that initial response, and we do see some, let's say, phenotypes like this, eventually all the tumors, all the mice succumb to their tumors. So then we decided, well, this is, let's say, not standard of care and so on. We know this model is like we're pushing the limit. So let's go back and take what is like the true and tested model, which is B16 melanoma cells treated with CTLA-4, the Nobel Prize winning experiment from Jim Allison. So for this, we engineered now in melanoma cells, a small and large deletion, put them in immune competence, subcutaneous is much easier, and then we just monoagent CTLA-4. Now, just to remind you, monoagent CTLA-4 in this model is not curative, but you do see a very strong response. So what we observed is that in our small deletion tumors, interferon proficient cells, we actually get the response as one would expect. And of course, in our large deletion, we are able to absolutely blunt this response. And again, this correlated with higher activation of CD8 T cells. So the status of the 9P locus alters response to immune checkpoint blockade. And now more and more clinical data is actually confirming this. Now, when clinicians make this correlation, and it's been there for a while, they say 9P loss. What we're modeling is interferon loss. So now we actually have a causal link for the type of, what are the genes responsible for these correlations? Now, full disclaimer, I've been talking about interferons a lot, but what I've been telling you is just I deleted different regions of the genome. So we haven't actually, until this point, functionally confirmed that interferons have anything to do with this. There are other genes in this locus. There's a lot of non-coding genes. So the model we have here that, well, this is very well known, that CDKN2A&B, blunt proliferation, and this is what we propose, that type 1 interferons activate CD8 T cells that blunt metastasis. So a prediction of this model was, well, if we play with interferons themselves, we should recapitulate some of these phenotypes. And for that, we do, as we did with the interferon, the CD8 depleting antibody, we have an interferon blocking antibody where we specifically rescue the metastasis of our interferon proficient cells without affecting whatsoever our large deletion. So at least we know that functionally it is about interferon signaling. Now, we do have a problem that I alluded at the beginning. Why? How does this even matter? Because in pancreas cancer in particular, there's almost as many immune infiltrating cells than tumor cells. And we know that every cell can make interferons, and immune cells can make more interferons. So is it a matter of threshold? So you have a bucket that you're filling up, and then when you remove one source, you just reduce this level of interferon? Or is there something specific Are we really maybe oversimplifying the interferon pathway and there is a deeper level of regulation? So I guess the question is, are all interferons made equal? And the way I came up with how do we solve this is, well, we have this fancy tool that I only use once in a paper about a method, so let's just use it again. And instead of making 2 deletions, I made 10, I'm only showing 6 here, that we increasingly eat into the interferon cluster. And then, of course, came what is absolutely a struck of just randomness, because I decided that I'm going to do an in vivo competition assay, where I'm going to mix all these genotypes, put them in a mouse, and see what happens. Now, up front, the prediction from this experiment is these are secreted molecules. If you put an interferon-producing cell next to a cell that doesn't produce interferons, since they're secreted, the interferons should be the dominant phenotype, and that is what we expected. Interestingly, that's not what we saw. What we saw is that the tumors came out in patches, so some cells were GFP high, and some were GFP low or negative, as we had seen originally in our immunoediting experiment. Now, at this point, I don't know what is what. They're all mixed together. I don't have no way to tell. And this is just a flow plot to show that we have this pretty cool bimodal distribution. We did this I think we did it in 10 different hosts, so there's a range of variation into this, but this is roughly how it looks. And what we did is, okay, instead of, let's go and backtrace. Let's just sort for GFP high and GFP low and see what is the genotype of these cells, and can we find anything that makes sense? And I'm just showing you one of the 10 tumors. The only common gene that had to be co-deleted for these tumor cells to evade immunity was just this first interferon epsilon. There are 7 papers in the literature talking about interferon epsilon in cancer, I think maybe 20 in total. It's a very interesting molecule, and we were pretty surprised about this because I really thought this would be a combinatorial effect of all interferons. So how can we actually reconcile all of this? I'm not showing, but this also holds true for metastasis. So if we compare in the pooled experiment the metastasis to the primary, it looks exactly the same. It's just interferon epsilon, at least in our system. So again, we go back to the idea. Is interferon epsilon the major interferon in our system? Is it tumor specific? How is it all working? So then what we did is we went to, of course, our interferon proficient tumors and sorted out tumor cells and immune cells, and just did a very basic, like, where are these things even expressed? Where are interferons expressed? There are 17 of these. I'm just going to show you a few. And what we could see is that, as the literature had been correct, interferon B1, which is the poster child for type 1 interferon biology, is very much expressed in CD45 positive cells. Now, when you go to the end of the spectrum, interferon epsilon is actually only expressed in tumor cells. So this might explain something. We just confirmed this genetically because these genes are very poorly expressed. And just to show that if we sort cells out, immune cells, of course, are not knockout. So the interferon B1 levels are there, but we effectively can't detect epsilon anymore. So at least we know both by expression and genetics that interferon epsilon is exclusively expressed by tumor cells in our model. The other thing is everyone in the field has been linking type 1 interferons to tumor response through the cGAS-STING pathway. This makes a lot of sense. Chromosomal instability will engage cGAS, which will activate STING. And interferon B1 exclusively is the major type 1 interferon that is part of that axis. So when people say cGAS-STING type 1 interferons, they mean interferon B1. So what we did is we went to our at least cultured cells. These are 2 independent cell lines of each genotype. And at baseline, as one would expect, because tumor cells, at least these ones, don't express interferon B1 at all, but when you give a STING agonist, DMXAA, you can actually now detect pretty robust expression. Of course, our large deletion serves as a negative control. On the other hand, interferon epsilon doesn't respond to anything. We have not yet been able to find a single molecule or pathway that engages. It just seems to be constitutive in our cells. There are some links with estrogen receptor, but at least our experiments perturbing the estrogen receptor have not yielded anything fruitful. But we know that at least in this system, interferon epsilon does not respond to TLR signaling or to cGAS-STING. So this already puts it in a different league than the other interferons. So now comes the question, right? How much can epsilon explain these phenotypes? And now this is going to be a bit of a sledgehammer experiment. So I'll walk you through it. It's a lot of data. And so how do you test if an interferon does something? Well, you overexpress it in cells that are knockout. That would be one way, and that's what we tried. The problem is these are extremely potent immune activators. So it's really hard to get a tumor to grow. So what we did is, okay, what we're gonna do is we'll make him inducible. So we have an RTTA activating the CRE3G, and it'll drive either a non-secreted, so it's exactly the same as full-length epsilon, but with no signal peptide, epsilon or B1, so we can have an inter-interferon comparison. So we engraft our tumors, let them grow. Once they reach a certain size, we activate the interferons with doxycycline for a week, and then we measure. Again, there's a lot of data here. I'll walk you through it. So the first thing is we have the primary tumor size, and we have two different genotypes. So the first thing we notice is that in interferon-deficient large deletions, both type 1 interferons were actually pretty effective in reducing the primary tumor size. The small deletion that has interferon epsilon doesn't really care. And I checked, this is overexpressed 1,000-fold, just doesn't care. But it does care about B1. When we look at metastasis, it follows a very similar pattern. We have a very strong effect. Well, these guys don't do metastasis to begin with, and the large deletion responds very well to epsilon, a little worse to B1, but on average, they both respond in reducing mets. So I don't mean to say that I understand or that we understand the biology of this. This is just to illustrate a point that I'm trying to make, is that when we look now at the molecules that are supposed to be affected by these interferons, I'll walk you through this again. The small deletion in dendritic cells, none of these, so epsilon does nothing. B1, if anything, reduces the number of dendritic cells, antigen-presenting cells. In the large deletion, only epsilon is able to attract more dendritic cells, and B1 has no effect. On the other side, CD11B cells, so monocytes and macrophages, B1 in small deletions reduces the numbers. In large deletions, it doesn't change. Again, it's not that I understand how any of this works, it's just to illustrate. As a field, we've collectively been referring to as the type 1 interferon signaling pathway. And now we have experimental proof in vivo. There was some biochemical data in vitro that the binding affinities were different, but now we actually have data in vivo that your genotype as a tumor cell will determine your response, and that your response to different interferons will be different. So there's a lot of, let's say, unknowns now. The other thing that we don't really understand is why in that competitive assay didn't the interferon proficient cells rescue for the others? What is the effective range of signaling of these molecules and so on? So at least these data argue that type 1 interferons are heterogeneous, not only on their regulation, but also on their effects. And I think that at least this is a lesson from genetics to, let's say, immunology, and I'm still learning immunology, that there's so much more to dig in. And I'm just talking about one cluster of genes in one deletion in one tumor type. So I'll just wrap it up there. I've been talking quite a bit now. And what we discovered in this paper is we don't not only develop this method to make deletions, but I think we actually uncover some very interesting biology about type 1 interferons. So we have a novel activity within this locus, which had been associated with very poor prognosis. Interferon epsilon had never been implied in anti-tumor immunity. And now if you just forget about the details of the mechanism, if you look at all the genes mutated in the TCGA or in any patient, if you look at the top 20 genes by point mutations, none of these are bona fide immune molecules. When you start looking at the copy number space, all the immune-related genes are starting to pop up. This genetic event affects 15% of all patients, of all cancers. So this is actually one of the major genetic routes for immune evasion, which for many years was thought to be non-genetic mediated. Now, interferons are really cool. Scott's really excited about it, so I'm not gonna work on them anymore. I have a lot of other questions I wanna work on. And I'm basically trying to, let's say, reach the title of this talk, trying to understand what do copy number alterations do. And the ability of Macheta now have expanded this to, instead of deletions to do other type of genetic engineering, we can start, the idea is to create de novo complex karyotypes. So we can actually understand, does the order of events matter? Do we have convergent or divergent evolution? Are there other mechanisms? There's cluster of interferons, but there's many other clusters of genes that are recurrently gained or lost in tumors, particularly related to immunity. What is the role of extrachromosomal DNA in tumor heterogeneity, and so on and so forth. So these are all the things that are, let's say, plaguing my dreams. And well, I'll just leave it at there, and I'll just recap. So the grand vision of what I've been doing. So in my PhD, I worked a lot in intratumor heterogeneity, working on stem cell phenotypes and whatnot. And now in the postdoc, I've been working on different genotypes across different tumors. So the idea of my lab is that we'll try to combine all of these two to really get into this very complex problem of tumor heterogeneity from the lens of copy number alterations. So now I will effectively wrap up with the most important slide, the acknowledgements. Of course, I want to thank my mentors, Scott Lowe and Cal Sanov, who is a fantastic collaborator who co-led this study with me. He's a master metastasis expert. All the other colleagues in the Lowe lab, there were maybe 30 people missing from this slide. Of course, our computational collaborators, Dana Pear, Ronan Roshan Noor, Fayaz, I already mentioned with Amy, that helped us with the COMPASS data set, our funding, and come visit me at VHIO. I'm always happy to chat and meet new people. So thanks so much for the invitation, and happy to take any questions.
Thank you very much, Pancho, for the presentation. It was great.
Thank you.
Before giving time for the attendees, a couple of very short questions. One is that when you look at patients, human patient samples, the deletion at 9p, how many times is short? How many times is long? And if there is any case that only affect the the interferon genes?
Yeah, it's a great point. So it's tumor dependent. In pancreas, it's 50-50, large and small. In GBM, it's 60-40. In mesothelioma, it's 80% small, 20% large. The interferon only, it's a statistical anomaly. There is none. So I do think that the driver event of the genetic deletion is the loss of the tumor suppressors. Now, in the paper, we actually modeled, well, now we can play with this. What if you just remove interferons? And what happens is you get that immune editing phenotype, so you retain GFP, but it's not sufficient to make metastasis. So for metastasis, you need to actually lose both parts of the locus. For that early immune evasion, you only need to shut down interferons.
And that is what that you mentioned just briefly at the end. So you're using now Machete to do new things, new deletions. So what can you do with Machete?
Well, the record is 45 megabases, but I'm quite sure I could get rid of a full chromosome. So the thing is, with a breakpoint, you can PCR it with a like aneuploid. I have to do FISH, and I just started the lab. I've been too lazy to check. No, so like, let's say there's many other regions, but let's say being at VHIO, I think I'm in an excellent position, and I've been having meetings with the clinicians because, I mean, at the end of the day, we do this for a reason. As well, one, to understand the biology of cancer, but to help patients. So there's a plethora of genetic evidence out there that copy number alterations are associated to poor response to different clinical trials and so on. And that wealth of data is not being explored functionally. So my view now is I'm basically knocking on the door of most of the clinicians. Is like, do you have any copy number profiles for me? Are there any interesting observations we can translate into our mouse model so we can like have mechanistic understanding? Because the corollary as well is we're trying to understand what these do, but there's a lot of these genes. So MTAP here is also a sensitivity. It's not a driver of any of the phenotypes, but it's been used in clinical trials. So I think with these types of approaches, we can like start to do this more systematically. So that's kind of my thing. I know I haven't given you any specifics, but that's kind of where I'm thinking.
Thank you. Questions?
Beautiful talk. I really enjoy it. So you focus on chromosome 9 for obvious reasons, but the question I have is how frequent is that the tumor, so a bunch of a whole chromosome of a tumor suppressor is co-deleted with genes related to the immune invasion? Do you know that?
So how many clusters of immune-related genes?
So whether, like, the question is, could you actually check whether zones where there is like a big jump of deletion coincides with other immune deletion elements?
That's the aim one of my lab, yes. So we are. So there are other, let's say, HLA cluster comes to mind. The interferon receptors themselves are in clusters. Most interleukins, and I just think this is like how genomes evolve, are just like copy pasting. There's a lot of tandem regions that are actually getting amplified and deleted. So in Down syndrome, that extra gain of 21, that's where all the interferon receptors are, and that's been associated to why they are more, let's say, protected to some types of tumors, but more prone to others. So I mean, that's hand wavy, but that's
And
what's
do
been
you know
put
if they're close to other tumor suppressor group of genes and this?
So that's the thing is, this is a rare case where the tumor suppressor is lost biallelically. So usually you get a point mutation and then a large chunk of the genome, and that loss of heterozygosity event is usually much bigger than what I was showing you here today. So we were kind of like hedging our bets with this one. So in the case of P53, you have, let's say, a point mutation P53, and then you usually lose a lot of 17p. Now, everything is immune related if you look far enough. So I guess the answer is yes. The possibility that a lot of these molecules are doing something to immunity is very high. Smoking guns, like a cluster of purely immune molecules, there's maybe 5 or 6 in the genome, but yeah.
Thank you.
So,
Oh.
cool work.
Thank you.
So from your last experiment, I get that interferon epsilon is working through dendritic cells probably to activate CD8 T cells, and
Yes.
do you know which specific phenotype of dendritic cells is being targeted by this interferon?
Yeah, it's a great point. I sugar-coated all the antigen-presenting data. Usually my audience is not as keen as, no. In reality, what happened is, so what we see in the immune phenotyping is we have effects on CD8 T cell phenotypes, but where the interferon signaling itself is most changed is in professional and antigen-presenting cells, so DCs, macrophages, and B cells. There's a paper from Bob Schreiber's lab from like 2011 where they knock out, it's in a model of sarcoma and so on, but if you knock out the interferon receptor only in CD11C total DCs, you blunt a lot of the immunity against that particular tumor. We didn't have those genetic tools at hand, but at least in our system, it's CDC2s that are the ones that are most changed. Like a lot of people have asked for the single-cell RNA-seq data to look at these dendritic cells because the type 1 interferons and DCs are a very, very hot field right now. Happy to talk more in detail if you want. I can like show you the exact populations, but we know it's CDC2s specifically. In so interferons did something different in every antigen-presenting population. So it changed the phenotype of DCs. It reduced overall B cells, so no cell state switches, just less of everything. And in macrophages, most of the states were the same, but a new state appeared in the interferon deficient ones. So we have it somewhere in the supplements of, this is like, they can do everything. So that's why I didn't go to, it would get too complicated, but thanks for the question.
Yeah, very, very nice. I wonder, I mean, especially coming from the Scott Lowe lab, so are you considering scaling Macheta up to a genetic loss of function screening approach to really?
Yes. So I have, if someone's brave enough to try it, I have some ideas how to get that. The major bottleneck here, and I'm sure you know this better than I, is that the knock-in is very inefficient. So if you want to do genome-wide, you always need that molecular handle because if not, small deletions always win to the big deletions. Yeah, so I have some ideas how to play around with this, how to increase the efficiency. Now, the other trick or the other issue is I don't, eventually maybe genome-wide, but you could imagine having a pool of cells with different knock-ins and then a pool of guides. Now, most of your cells will die because the chances that the 2 guides that hit the right cell with it, so it has to be a very focused and with enough numbers you can scale, but the combinatorial gets a bit crazy. So I'm thinking more on the lines of improving the efficiency of the knock-in so it's easier to make. And then probably you could actually do tiling. So you can, in one particular knock-in line, you can, let's say, anchor one guide and then just create a sweep. Yeah. So instead of what I did one by one, you could do it pooled. I haven't tried it, but that should work.
Just because you already got this question. So is there something specific about immune genes that they tend to be more in clusters than other genes? Or is this
I think most genes tend to be in, like now my recent obsession in life is whenever I go to a talk, I go and look, oh, this gene is in this place. I go and look at the genome browser. So histones are also a very famous cluster.
Yeah.
So yeah, I guess
histone variants.
Not the variants. That's true. Yeah, we should talk about that though, because I don't know what would happen if I knock out the histone cluster. I mean, it's complicated, but
Die.
Yeah, exactly. But it's heterozygous, so.
Dice aquí está en heading paper.
I want to thank you for the talk.
Thank you.
It's wonderful. I have just a question regarding the CRISPR approach.
Yeah.
So if you have checked, for example, for chromotripsis, when you trigger the big deletion, you have other alterations that are not only the one that you are looking for.
Yeah, so we did, like when I do the sparse whole genome, see, I'm just showing you the plot because if I show you the whole thing, you don't see the deletion. I was really concerned about that, and not even about chromotripsis, just by drift. Our cells are p53 knockout, so I thought, well, we'll have a very short window before I passage these cells 2 or 3 times, and then all the genomes are a mess. We put them through mice like 3 times, and they really like this genotype. We have noticed, so there is some noise, but it's not like segregating with the deletion. So as far as I can tell, the only thing that really happens is you make this deletion, but yeah, we haven't done it single cell, which would actually answer to your question because we don't know. I've done single cell clones just to validate the frequency of the event, but I didn't do single cell whole genome sequencing of each of the individual clones. So it's a very valid point. As far as we could tell in our system, that wasn't a big issue.
Thank you.
So very nice talk. I enjoyed a lot.
Thank you.
So maybe you mentioned, and I'm just asking something that you said, but so what is the specific signaling about this interferon E gene? So do you know anything about the subset of genes that are being specifically regulated? Because I guess it's a signaling that at the end ends in a
program. Yeah.
Yeah.
So you mean the downstream effectors? Yeah. So what
makes it different?
Yeah. So the only thing we can tell, and this correlates well with the published literature on let's say just the biochemistry of the receptor with the ligand, is that epsilon has less affinity than B1. When we've looked at, let's say, canonical ISG targets, they can all engage them. So we always thought, oh, maybe it's a second layer that now you access a different transcriptional program, and this is why it's different. We have not been able to find anything like that yet. I don't know that we're doing the experiments in the right way to do this, which is we have our cell lines and we treat with recombinant cytokines. But yeah, we're thinking along those lines. The answer is we just don't know. So far, it seems that they do the same, just one's more potent than the other. So it could be something about chronic or acute.
Just a very stupid question. So in your method, do you think it could be used to play a little bit with repetitive regions?
Yes.
Like adding like electrochromatin, I think copies, whether you could really have a specific strong output in the general stability.
So we've thought about, or I've thought about those type of experiments. The major issue we encounter is where do we begin? We need, if you have a solid readout that we know, we know that, because retroelements are everywhere, so which ones would you delete? Which region would you want to delete? And if you have one that you know functionally has a phenotype, then we can start playing around with that to dissect it. Right now I've been reluctant because it's which one first? So I love the idea because at the end of the day, I'm gene-centric at this point, but there's a lot of things you can study about chromatin configuration because people like TADs and so on. Well, now we can just get rid of all of them and see what's the effect on transcription and so on, 3D genome and retroelements, of course, are a big question there. It's just a grand challenge as well. It just came out the other day. So I like the line of thinking. I've been hesitant because I don't have a good experiment to do yet.
Thank you.
Okay, thank you. Let's finish here. Thank you very much.
Thank you very much, everyone.